Hi, my name is Monica Lawton.
Hi, I'm Brittany Montgomery.
Hi, I'm Noor Mustafa.
And we are presenting our experiment on the effectiveness of terminal cleaning and on minimizing microbial contamination for microbiology for Dr. Lon Bloodworth. Our study aimed to examine the effectiveness of terminal cleaning on microbial contamination on nationally recognized high-touch hospital room surfaces. We hypothesized that the terminal cleaning of the 5 high-touch surfaces identified will not ensure 100% microbial removal. To test this hypothesis, samples from 5 high-touch surfaces were retrieved via swab culturing before terminal cleaning. To evaluate the terminal cleaning procedures, swabbing of the same 5 high-touch surfaces were swabbed after 4 randomly selected housekeeping staff members on 4 randomly selected hospital rooms at St. Vincent's Medical Center, Southside, completed their cleaning. The samples from each of the 5 surfaces were grown on universal growth media to evaluate the cleanliness. After 26 hours of incubation on the TSA plates, the microbial growth was quantified by colony-forming units. The results illustrated significant decrease of the contamination of the 5 high-touch surfaces after terminal cleaning, and further testing would aid in indicating a larger trend that has been witnessed in this study.
Microbiology is often taken by students anticipating careers in the healthcare or medical fields. This course sets students up for success by introducing new techniques and skills that are often not taught in a classic classroom or lab setting. Microbiology, in summary, introduces students to microorganisms and their attributes that most have only heard of and the proper way to handle and exterminate said dangerous microorganisms. While microorganisms are active everywhere, the precautions that are needed to be taken within a medical field to prevent the spread of serious, harmful microorganisms are not to be taken lightly. Healthcare-associated infections a patient can come in contact with while receiving medical treatment in a healthcare facility, are a major yet often preventable threat to patient safety. Hospital cleanliness and proper environmental infection control strategies can effectively help to limit and prevent disease transmission. Terminal cleaning is the cleaning methodology used to eliminate environmental pathogens in a patient's room after a patient's departure to adequately prepare a hospital room for a new patient admission. The cleaning of hospital rooms and open areas is extremely important, not just for the new patients who will occupy the rooms, but for the hospital as a whole. In a study performed by Husledge, the most explicit high-touch areas have been identified as bed rails, bed surface, supply cart, overbed table, and intravenous pump.
Our objective with this experiment was to evaluate the efficiency of hospital terminal cleaning procedures by using agar culture testing to quantify pre-cleaning and post-cleaning aerobic colony counts. We got our samples from St. Vincent's Medical Center of Southside. They utilize the Touchpoint Support Services, which is a contracted service
to perform the housekeeping needs of the facility. The surfaces we chose to test were those identified as high touch in the study earlier cited. What we did in our experiment was choose 4 soiled hospital rooms at random. Immediately after the patients were discharged, Medline's sterile cotton-tipped applicators were used to sample 5 high-touch surfaces. The surfaces were sampled and identified in a study by the Society for Healthcare Epidemiology of America, based on 50 interactions between 50 healthcare workers and their patients. The surfaces identified were those that were frequently touched during the patient and healthcare provider interactions. MedGuard nitrile non-powdered non-sterile gloves were used to avoid contamination of the samples. A sample area of approximately 10 centimeters by 10 centimeters was selected on each surface, and a sealed, sterilized Medline cotton-tipped applicator swab was moistened with sterile distilled water, then rolled firmly across the sampling area. The swab was returned to a clean, labeled biohazard bag, which was sealed for immediate transport to the appropriate laboratory housed in the Florida State College at Jacksonville Deerwood campus. This procedure was repeated on the remaining surfaces and performed in each of the 4 rooms. A clean pair of gloves, a new cotton swab, and a clean biohazard bag were used for each sample to avoid contamination by foreign microorganisms. This resulted in 40 separate swabs in 40 biohazard bags. Terminal cleaning procedures were then performed by 4 randomly selected touchpoint housekeeping staff members on the 4 randomly selected hospital rooms. All 4 staff members involved in the study had been trained to follow the same standard cleaning and disinfection protocols as outlined by Touchpoint's award-winning onboarding and training program. Each housekeeping staff member was provided with the same cleaning equipment that had been tested and in place by Touchpoint Environmental Services. The housekeepers were not notified of the study to maintain the integrity of the investigation. 10 minutes were provided after the cleaning products were used to allow maximal disinfection potential. After this time, the same 5 high-touch areas were swabbed and labeled disinfectant samples were sent to the lab. The same procedures were used for the post-cleaning samples that were used for the pre-cleaning samples. And we also returned the samples to the lab.
Proper microbiology laboratory safety rules were practiced as followed. The universal blood and bodily fluid precautions were taken due to the possible presence of blood-borne pathogenic microbes housed within the samples and within the hospital bedrooms. TSA plates were chosen as the growth media unto which the samples were inoculated, utilizing aseptic technique close to the flame. TSA was the media of choice to fulfill the objective of isolating and observing the colony morphology of the microbes present in the sample. All surfaces of the cotton swab applicators were rolled onto the surface of the agar plate to ensure the entire collected sample was collected near a flame to ensure sterilization. Special care was taken to prevent the tearing of the agar. TSA plates were then labeled with initials, the culture source, and the date with a line drawn directly down the center of the plate to distinguish the pre-disinfectant and the post-disinfected samples. The samples were then incubated for 26 hours at a temperature of 37 degrees Celsius. After analysis, the plates were then discarded in the designated biohazard receptacle for sterilization. Following the incubation period, the samples were removed for immediate visual analysis and examined for growth. All 3 disinfectant agents resulted in decreased microbial growth on the non-porous surfaces tested, with few exceptions. The results viewed were observed and based off both the before and after samples of each of the 5 high-touch areas in 4 different rooms. Following each of the results of the samples in our paper, you will see a image of all of the samples in order, and then also a bar graph of each, illustrating the amount of growth before and after of the plate on a scale of 0 being the lowest and containing no visible growth, and 5 being the highest with very heavy visible growth. The results of our experiment were conclusive and illustrated human flaw shown in patient room 1, surface 3, and patient room 4, surface 4, showing foreign microbes being introduced.
Conclusively, the amount of microbes present with an unsanitary room on the 5 high-touch surface points dramatically decreased after terminal cleaning. This data collected supported the hypothesis that there will be microbes remaining on surfaces after terminal cleaning. Respectively, the experiment was anticipated that human error would occur. During the sampling of the surfaces, the same 10 by 10 area was swabbed in each room. We did this to prevent choosing a different sample than was used in the previous study. Based on the findings, surfaces in their entirety were not 100% clean after terminal cleaning. While there are terminal guidelines enforced, it is up to the housekeeper's discretion to maintain the level of sterilization needed. This experiment only conducted samples of the 5 high-touch areas listed. It did not do a thorough investigation of the cleanliness of the overall room. In addition, the contact time on the labels of most registered EPA disinfectant agents are too long to be practically followed in a healthcare setting. We think this might have contributed to some of the error in our results. From this data, it is important for patients, staff, and caregivers to realize that despite standard terminal cleaning practices, the environment tested may not always be effectively sanitized due to human error. Further testing is recommended to examine the effects of increased exposure times as well as the specific microbial species that were able to overcome the disinfectant use.
With more testing, we will also be able to see trends between each of the surfaces.
The sources we used for our paper are the Center of Disease Control and Prevention, as well as our Lammert Techniques of Microbiology student handbook.
Thank you.
Thank you.